so that, people that they would be here, they can get access to those notes. And if there are questions, after people look at those notes, those who are not here, then they can send the questions, as we said yesterday, through the class president. And I can get responses back, and those responses will go to everybody. Okay? Okay, so we started out yesterday talking about the anatomy of bacteria, so just the way you have it kind of listed on the syllabus that you have. And so I mentioned yesterday that it would take, a couple of meetings to talk about the anatomy of bacteria, to kind of give us terms that we can use as we begin to look at what's going on inside the oral cavity. And so for some, a lot of the things we're talking about is review. For others, all of it is new. And we're just wanting to make sure that we're all on the same page. And so for today, we're still looking at the anatomy, and we're looking at the relationship that this has to pathogenicity. And so some of the concepts that we want to kind of cover today include, how bacteria are organized. So yesterday, we kind of reminded ourselves that we have, gram-positive, gram-negative bacteria, we have gram-variable organisms, and excuse me, and then it's one of those gram-variable organisms, acid-fast positive, and we mentioned mycobacterium as one example. And we also mentioned, some additional features that distinguish gram-positive from gram-negative cells, and we'll see more of that today. The second point, the bacterial cytoskeleton. And so one of the things that we kind of want to convey today with this is that a lot of the research that's ongoing to understand what bacteria have in them, essentially, things that have homologs, in eukaryotes, is the cytoskeleton. And what's, been found is that we have homologs of actin, intermediate filaments, tubulin in these are bacterial cells. And so we'll see examples of those. And then we want to look at the cell membrane or cell envelope of the cells. And we want to make sure that we're aware, okay, as we mentioned yesterday, with the gram-negative cells, right, we have the plasma membrane, but we also have that outer membrane. Everybody remembers that. We have the periplasmic space. And so this kind of differentiates the gram-negative cells from gram-positives. We're going to look at the peptidoglycan, talk a little bit about how the peptidoglycan is synthesized and why it is important to know this, because a lot of the the drugs that are aimed at bacteria, are directed to the cell wall. Okay, so if we can disable the cell wall, then the cells will not survive. Excuse me. And then we're going to look at some of those things that are targeted by enzymes, by antibiotics, and look at some of the proteins we have in the cell membrane. And in particular, a major takeaway today is we want to really know what LPS is. So we want to know the structure of LPS, just the general organization of LPS, because of the importance of the O antigen, okay, that's used for serotyping and typing bacterial strains, and the importance of the lipid A, okay, which is what can be a problem, when that's shed in dying cells and it's in the blood. So we want to start here. Excuse me. And so we want to understand the following. So pathogenic organisms, pathogenic microbes. So the term, pathogens is used to refer to the various microbes that are associated with disease. And so, whenever I mention this, I always immediately add that the microbes are not just bacteria. Okay, so pathogens, can be bacteria, they can be viruses, they can be parasites, fungi, etc. So we want to remember that. And so, the mechanism that a particular microbe will use to cause disease, right? We have the pathogenesis. So we describe the pathogenesis of different infections and so on. And so if we have replication of a pathogen, in or on the host, that refers to the infection. And so we can have, specific signs and symptoms that are associated with specific diseases. And so, in many cases, you have etiological agents that once they establish an infection within the host, and as, the infection progresses, and in many cases, once the immune system, starts to respond to the presence of antigens, from the pathogen, there are particular signs or symptoms that will develop that in some very specific cases, those signs and symptoms will be pathognomonic. They will be very specific for that infection, so that if you see that sign, it's caused by this organism, okay? And so those become important. Now the table at the bottom just kind of gives some signs and symptoms associated with some infectious diseases, and we have a mix of viral, plasmodium or protozoan, and bacterial type infections. So just showing that in either case you're going to be able to see signs that are representative of those infections. And so it's important to note that not all pathogens have the same ability to cause disease. And so we talked yesterday a little bit about, frank pathogens and opportunistic pathogens, right? And so, the ones that will produce disease in immune competent individuals, right, are referred to as primary pathogens. So a person does not have to be immunocompromised, for an infection to set in. So in those cases, we have, again, healthy individuals, intact immune system, but they're susceptible to infection. We have primary pathogens that can do that. We also have opportunistic infections that can take hold, and it's important to also distinguish amongst those. So we can have infections caused by organisms, and the one that I always like to use as an example is a protozoan, and you might have heard about it. This is Naegleria fowleri. Anybody heard that organism before? Naegleria fowleri. Anywhere? Okay, so this is an organism that we have, it's in the soil, in water. I mean, we're always around it, nothing happens to us. But if we happen to have the organism forced through the nose into the brain, like when diving or swimming, things of this nature, we splash in water, children do this, then the organism gets inside the brain and causes fulminant infections, and can kill within 48 hours if it's not if there's no intervention, and that's pretty much when it's found. It's found at autopsy. So in those cases, individuals are healthy. They happen to get the organism in there, and this is what happens. Go ahead.
Does it have to be forced, or just like inhaled from the soil?
Inhaled can be, and so inhaled would categorize as also being forcibly getting things in. Yeah.
What species is that?
Fowleri, Fowleri, Naegleria. So this is an amoeba. Okay. So that's an example of an opportunistic pathogen, okay, that can infect healthy individuals. Okay, so individuals don't have to be immunocompromised. And so when we think of opportunistic pathogens, right, we think of immunocompromised states. And another way that we can have organisms, opportunistic, that can cause infection is if they get into sites that are not normal for them. So regular commensal organisms that we have in the body can cause infection if they get to the wrong place. Okay, so if we get the cells into the blood, right, we can have infections there. If we have bacteria that travel from the mouth, they can end up in the heart. They can be around the heart valves. We can have inflammation there and so on. Those are examples of other, opportunistic type infections. So you have a list of organisms. So you look at, these are some examples. So you look over there at the primary pathogens. We see, a number of bacterial species. We see the diseases that they cause. And then below that, we see the opportunistic pathogens. Again, we see a mix of both gram-positive, gram-negative bacteria and the type of, infections that they cause, all the way down to the bottom over here where we see Streptococcus mutans. Okay, we said we're going to see this name over and over again. And so again, we see the tooth decay, and this is one of the organisms that's of most concern, leaving the oral cavity and getting into the blood and being around the heart. And so we see that. So just take a look at that list later on. just to kind of familiarize ourselves with some of the organisms that we have on there. And so, we looked at this yesterday, and I just wanted to kind of have us remember again that, we need to know what Koch's postulates are. And so yesterday we talked about some of the situations where the postulates will not exactly work 100%. If we don't have the right hosts, to grow the pathogen in, we have difficulty. If we don't have cultural conditions, to show, that's going to be a problem. But in general, if we have an infection and we have it suspected that the organism is the one causing it, we should be able to isolate that, right? Put into clean animals, show the same infection, able to isolate the organism from there, put them in plates, and be able to show growth, okay? And so the original intent of the postulates was to be able to use this to authenticate that we have the correct etiological agent, okay? And so we need to remember the postulates as they are. And so we need, so we have a bunch of terms in this next 2-3 slides that we just kind of want to pay attention to. So we need to be able to distinguish between infectious and non-infectious, right? So, this is different from things that are communicable, which are non-communicable. Communicable things, if we have droplets in the air, individuals are around, and if they breathe those in, for example, they can get, infections from those. And so in that case, we have something that's truly communicable. We can have stuff that's infectious that unless there's direct contact, exchange of fluid, penetration of the skin, that kind of a thing, introduction into the blood, for us to have infection, okay? So we need to differentiate those. And so those things that are communicable just by, being in the same environment, also we refer to as being a contagion. So we have a contagious situation. So you don't even have to be in direct contact, okay? As long as the organism is available in the environment, individuals can get infected that way. So we just want to kind of clarify the terminology. And if we have pathogenicity, and this is again not specific to bacteria, we can talk about the same things, with protozoan organisms or juveniles of helminths. So we need to notice that we can have adherence factors that are produced or secreted by organisms. And we'll say this a few times, just to help us understand the significance of this. And we can also have enzymes that are produced by organisms that allow them to be invasive. Go ahead.
Could you give me an example of non-communicable?
So non-communicable infection would be something, again, just comes to my mind because I work with that all the time, with malaria. Okay, so you have to be bitten by a mosquito to get the infection. Right? So, just by being next to somebody who has malaria, you won't get malaria, okay? And, another non-communicable directly, something like, HIV. I mean, you have to actually have the virus, in you just by being next to somebody who has HIV doesn't mean you're going to get HIV. So those are non-communicable situations. They're infectious diseases, but they're non-communicable.
So what's the difference between communicable and contagious?
So contagious actually, sometimes it's used interchangeably with communicable, but the contagious really means, so like if you have, a virus that you can have in droplets in the air, and if you happen to be in the environment with somebody, right, then you can get that. Or something like measles, okay? So if a person that has measles is in the same location with others, then the chance of having been exposed is pretty high, okay? Right. So does that make sense? So these are terms, so again, we use them just to kind of be clear in what we're talking about. Okay. All right. You had your hand up too.
Once the pathogen lands on the skin, of course, there's movement, stem cells fluffing off, existing bacteria, all this. How long does it have to release that hyaluronidase and get through? Are we talking seconds?
It depends on the
minutes,
organism.
hours?
So in some it's within seconds of being in the environment.
Okay.
So once they're in the tissue, and usually those that do that are invasive, right? And so the 3 examples we have on here are pretty important. So we have, in the first instance, so we're looking at the pathogens, we're in the epithelium, right? So we can have organisms that can, again, just think about it. So, and we can use the mucosa as an example. We can even use the skin as an example. So you can have organisms, penetrate, to the mucosal layer, but they can't get into the laminar prophet. Okay, and we can think about your histology, right? So we can be on the surface, of the epithelium of the mucosa, but we can't get to the basement area. So to do that, the cells either have to be able to get into the epithelial cells, okay, and kind of work their way through the basement membrane, or they will go through the lateral areas of the cells. They kind of go through the areas where you have the junctions to go through, okay? And so for them to be able to do that, they have to be able to produce substances that allow them to breach those environments, okay? Yeah, so that's what we're seeing, and that's the example we're seeing over here in terms of what organisms can have. Go ahead.
So highly virulent organisms evolve quite a bit of this.
Right, so they have
How do they
substances
know?
that allow them to do so, yeah.
It's not energy efficient to release it all the time, so how do they know, okay, I've landed on something, now it's time to release it?
And there are many questions surrounding that, in terms of, timing, when do they know, to do that. And, in some cases, it's fairly instantaneous once they're in contact with the substance that needs to be cleaned. So, right, there are a lot of studies that look at that. So the second example that I wanted to mention to you is that you can have organisms that penetrate the epidermis of the skin, but they can't get through the stratum basale. They can't get into the dermis area. So they can wander around, okay, that basal area, okay, and they can end up, dying there or whatever. And as they begin to release substances, as they're dying, then, we can have stimulation of cells, within the dermis responding to that. And in the epidermis, we can have, Langerhans cells begin to respond to them, okay? And they can drain those antigens and get into the lymph nodes, for activity. So, the questions around that are of great interest because you do have pathogens that are deposited into the skin, and they end up in the blood and are taken to various sites in the body. And many times the question is, how are they doing that? Right? So we know where they end up, but the question is, how are they getting there? And, especially if they get to a location and it's like a final location, right? So there's a lot of interest in research in this area to understand, like as you say, timing, concentration of the substances that are produced, how many, okay, and so on, yeah, okay. And so we have the first example there where they have on this, again, getting things, through the epithelium. And then we can also have, coagulase in bacteria, pretty important, especially, in things like Staphylococcus. As we're going to see later on when we talk about how we differentiate the cells, the ability to produce coagulase by strains of Staphylococcus aureus allows us to differentiate them from other species of staph, okay? And so coagulase is very important. When it's produced, it will act, fibrinogen, we'll get fibrin produced, we get this, clot that forms, and it basically allows the, organisms to kind of be together and kind of try to get away from the immune system. But we can also have, certain species of strep that can produce streptokinase, and they can break, blood clots that are formed, and essentially allow them, to get the bacteria further into the body. Okay, so these three examples are examples, of where having these molecules produced actually aids the bacterial organisms, become more invasive in where they are. And so we also have other things to think about in terms of opportunities and opportunism. We have the immune system, fail if we have, for whatever reason, we're not able to either phagocytize or phagocytize, we cannot process, and we're not getting, the antigens, to the T helper cells and so on. Any number of those things can affect, immune activity, and we can have post-defense failure. We already mentioned we can have microbes in unusual locations, and if we have, the dysbiosis we talked about yesterday, where we, affect the populations of bacteria, we can also have problems with that. Okay, any question? Oh, okay, yeah. So, no question? Yeah, all right. Okay. And so, we have a number of things to keep in mind. So host susceptibility and resistance to infection is determined by a number of things, not just the host immune status. Okay, so the age, there are other illnesses, chronic conditions, other infections, the epidemiology of that particular infection, the environment. If you have things that are transmitted by vectors, the availability of the vectors, the range of distribution of those vectors, any of those things can affect the ability for transmission to occur. Also, having virulence factors is important. So, we have strains within species of organisms from bacteria onwards, right? So we can have this, in fungi, we can have this in protozoans, in helminths. We have strains where, you're not going to see, pathogenesis, by their presence. But then, if they're expressing virulence molecules, right, and those are indicated as virulence markers, then, those can now allow, the disease-causing ability of that particular strain, to be seen. So, I guess exposure to the pathogen, is important, and exposure in the correct way. So if we have organisms that penetrate the skin, if that's their mode of entry, other ways, for example, ingesting them may not, cause, pathogenesis to occur, and so on. So again, we have frank pathogens, true pathogens, and then we have the opportunistic organisms, okay? So frank or true pathogens, opportunistic pathogens, so we need to remember those two. So when we talk about infection, and this is going to be important when we look at what's going on inside the mouth. So in cases where we know the single organism, okay, the etiological agent, okay, this single organism is going to cause the infection if it is in the host, right? So in that case, we're basically dealing with that one organism. But we also have situations where multiple organisms essentially, can give the same, clinical picture of the infection. And so in those situations, sometimes, diagnosis, can be difficult, kind of trying to figure out what's going on with the symptoms can be difficult. You have to be able to establish and show, that the organism is there. So with bacteria, you need to be able to show that the bacteria is there, maybe culture it, show that you have it, in the culture plates. Do the gram stain, show that, it is indeed, a particular gram staining type and so on. And so we can have those. So when we look at the single situation, so when we have Clostridium tetani, for example, right? We have tetanus, we know that's where it's coming from. If we have multiple organisms, same picture, we might see, meningitis, we might see pneumonia, you can have multiple different organisms showing the same thing. And so, and in some cases, we have, we're looking at something like pneumonia and meningitis, we may have that, be from bacteria, it may be from virus, it may even be from parasites. And so again, being able to diagnose correctly is key, okay, to know what agent is involved. And so when we look at disease stages, we have incubation periods, we have the prodromal periods when we begin to have, the symptoms, showing up. We can have an invasive phase where we now have the organisms, within, deep into the tissues. And, the immune system, has already been activated, so we have immune responses already going on, right? So initially, innate immune responses, they're followed by acquired, immune responses and so on, adaptive immune responses. And so we might then begin to see a decline, okay, as we're clearing the pathogen, right? And then, we have, a convalescence period. And so in some disease states, right, we don't quite have a nice clean appearance of these stages, and things can progress into being more chronic, okay? And so it might take a while, before we have any kind of resolution. But so, so generally these stages, will take place. And we can define these very clearly. In fact, that's the examples that are always usually used, for somebody, having, like the common cold. You can see the whole set of stages until, you have a recovery and, and people begin to feel much better. So, we want to know this. So, amongst the bacteria, we have a number of features that we're going to look at. But before we start looking at what's going on with the cell wall, cell membrane, etc., right? We just have a little brief set of features over here on this table, looking at what bacteria can produce in terms of the toxins that they produce. So we have endotoxins and exotoxins, and really the common endotoxin is the one that we have associated with gram-negative bacteria, okay, with the LPS. And so, if we're going to see in the structure, we have the O antigen, right? We have this core region, where we have, sugars associated, then we have this fatty acid, tail component, the lipid A. And so when bacteria, gram-negative cells, when they die, they release, this LPS, from the outer membrane, and it can, result in a number of effects in the host. And so all the way, to septic shock and so on, very serious situations. Now, we don't have an LPS, lipopolysaccharide in gram-positive cells. So gram-positive cells will secrete, exotoxins. Now, there are a few exotoxins gram-negative cells will produce, but we're always associating the endotoxins with them because of the severity of, what happens when we have that present. So the exotoxins associated with gram-positive cells, these are usually, protein, macromolecules. They can also have some very, serious effects, once they're produced, and we'll see a few examples of that. And they can target tissues in some very specific ways. And typically they're secreted. So unlike the LPS that's released as, in large amounts when the cells die, the gram-negative cells, the exotoxins are secreted, by the gram-positive cells. And so we'll see differences in how, how these two things work. So for now, we need to be able to differentiate, okay, endotoxin, we're thinking gram-negative cells. Exotoxins, we're thinking of gram-positive cells. And if we mention the endotoxin, we're thinking about this molecule that has the O antigen, the core, and the lipid A, which we'll see the structure of in just a little bit. And so the exotoxins, are important when they're produced. And so we have, the hemolysins that are produced by, by bacteria. And so on blood agar, we can actually see when these are produced. And so when we have the, go ahead.
Will LPS always prompt an immune response, even in a benign species?
Right, yeah. Its presence will elicit an immune response, yeah. So in fact, a way of thinking about it is that the LPS, is mitogenic. Okay, does that mean anything? When a molecule is mitogenic?
Causes cell division?
Yeah.
Okay, yeah.
Okay, so which means, its presence, will elicit this response, and cells will respond to it. Lymphocytes, will respond to it, by dividing. Does that make sense? Okay. Any questions about that? That's really important. Okay, because we have the gram-negative cells, in the mouth. We already mentioned, we keep mentioning, buffer monas, like yesterday we kept saying that. Right, and so we'll see, part of what happens when we elicit this immune response as a result of being in the oral cavity, and then the type of cells that are recruited as a result of that. Okay, so just kind of hold that thought. Okay, yeah, okay. All right, so we have, the different types of hemolysis that will occur when this is produced. So if we have complete hemolysis, okay, so we have beta hemolysis, right, and this is what we see, clear, and then we have, this alpha hemolysis, kind of greenish, incomplete, and then, so we want to be able to differentiate them, okay? So, so if we isolate cells, so remember how we talked about this yesterday, so yes, we have, a good number of cells that we cannot grow, okay, and we can identify the bacteria using molecular means, but then for those cells that we can grow, okay, so we're dealing with things like streptococcus and staphylococcus and things like this, the gram-positive cells, and they're producing, these exotoxins. So an example of an exotoxin is hemolysin. So when we grow the cells on the blood agar, and we allow the cells to grow, they'll hemolyze the blood, and so we look to see, okay, do we have complete hemolysis? Do we have a partial hemolysis? And then we can categorize them into the groups, okay? And so, again, go ahead.
Did you say that alpha hemolysis is this one.
Right, the one on the greenish, right, and the beta hemolysis, the here, yeah, okay? So we want to kind of remember that, all right? And so, go ahead.
Is small hemolysis typical of all exotoxins or just?
No, no. So this is one example. So this will, one kind of exotoxin is hemolysis. Okay? Make sense?
Yes.
Okay. All right. So we have a couple of questions just to kind of see the things that we've been talking about. All right. So the following feature is characteristic of eukaryotic cells. Okay. So we have 4 choices there. So which one of those is a eukaryotic cell feature? Okay, the 80S ribosomes, right? The 70S, bacteria, archaea, okay? So we have that. And so we mentioned the number of cells yesterday, and we had the tables, right? So which of these is not a common member of the oral cavity? Okay, everybody in agreement? Okay, so D. So the other 3, right, we're going to find in there, mutants, we'll find in there, buffer monas, we're going to see, the role it plays and so on. Okay, and so we just looked at the hemolysis. So if we have clear hemolysis by bacterial blood agar, which one is it? Okay, beta hemolysis, right? So the partial hemolysis, the green, right, that's the alpha hemolysis, okay.
So clear is complete.
Right. So let's go back to the picture. Okay, so what you're looking at, okay, are these areas of hemolysis around the colonies. Okay? Versus here, where it's incomplete. Right? Okay. And so this is important. So we're going to find out in, I think, next week, probably Tuesday, right? We're going to talk about the tests, that are done. Okay, so, and this is important because, again, it's not every, routine visit that a person makes to the dentist's office where you have cultures being, taken, right? So you're not going to, swab for cell growth every single time. But there are going to be situations where that might be necessary. And so the things that we're going to be talking about is what's going to be done. So if you're going to grow this stuff, the most common medium you're going to grow things on is just nutrient agar, okay? That's the most common type of medium. But we have blood agar, we have, differential media, McConkey's, we have all different ways, selective media, that allows you to kind of, get rid of the things, that are kind of in the way for you to get to the diagnostic, organism that you want. And so we have all these different media that are used, to allow us to do this. And one such is the blood agar. Okay, we're looking for this hemolysis to be present. Okay, so, just go ahead.
Sorry, what are the characteristics of epsilon hemolysis and gamma?
Right, so those ones, again, pretty much like the, no hemolysis, is really what those ones will show you. And certain degrees, of incompleteness, right. So, the one where you see, fully hemolysis is that beta one, the one that we had on the
But
on the right.
the epsilon and gamma also exist.
Right. And so the one, so of the number in there, the 3 you will see most commonly mentioned would be the alpha, the beta, and then, what's it, gamma in there, those ones. But the other ones, you can't do no differentiation, right? But with the, and then the 2 most common ones would be the beta and the alpha. Okay, yeah. So I just have these terms over here. Okay, we're not going to go through every single one, but some of fairly self-explanatory. But we want to look at them, and I'll just mention a couple of them that we really kind of want to focus on. And so one is the sequelae. So when we have a given infection, and so after that infection has been resolved. Right, and so there can be a consequence, following that infection that we will see. Sometimes, not too long after the resolution of the primary infection, but in other cases it can be, weeks, months, to even years, okay, before we see the sequelae occur. The other term in here that I want us to kind of think about is the last one, systemic. And this is because, you know, we can have, you know, local infections, local site, local tissue. We have the infection taking place over there. And then we can have a dissemination of the organisms to other areas of the body. Okay, and so we have situations like that. You know, one of the ones that we're supposed to address as we talk about bacteria in this course is, you know, when we have organisms that, you know, leave the mouth, you know, get into through the bloodstream, they end up, you know, in the heart, right? And so again, you know, we have the infection started over here, the presence of the organisms, but now we're concerned about the heart and other locations in the body. Okay, so the focal, local infections can lead to this systemic situation where now we're being concerned about, you know, effects on the heart, effects on the kidneys, okay, effects, you know, in other places in the body, okay? So we want to kind of keep track of those terms. And so, and I think we're all, you know, clear that symptoms and signs are different from pathogenesis. Okay, so those are so sometimes, you know, kind of mixed together, but they are different. So again, look through the terms in here and go through them. Additional terms in here that we need to know, and we'll use them as we need them. But the one in red, which I want to highlight today, that for the most part, the things that we're going to be talking about in the mouth are mixed infections. So we don't have just one single organism that's, you know, responsible for caries or responsible for, you know, periodontal disease and so on. And yesterday we kind of mentioned that. We said we will see Porphyromonas gingivalis. A lot of people study that organism. If you look in the literature, there's tons and tons of papers on just that one organism. And many of the research questions, you know, deal with, right, what are the other organisms interacting with Porphyromonas gingivalis, right? And so, and this is because, you know, many times you want to know the contributions that the other organisms have to the condition you're faced with, right? And so mixed infection is what we're going to be encountering, okay, in the different things that we're going to be talking about. So again, you know, the other terms over there, you know, please go through them and we'll use them as we need them. Additional thing over here, just to kind of mention, so how do we collect samples? Yesterday we talked about the fact that the plaque on the teeth, you know, can be examined for organisms, right? And not just in individuals, you know, present, okay, you know, going to the to see the dentist. But I mentioned to you that we have a lot of studies that are looking at, you know, plaque, you know, deposits and deposits of food items from, you know, 1,000 years ago, okay, you know, 2,000 years ago, and trying to understand if we have the same organisms, okay, that we see today, okay, and so. We can have things collected, you know, samples in different ways, and so depending on where the infection is located, right, we may be collecting, you know, sputum, you know, stool samples, urine, blood, you know, whatever, swabbing, you know, various areas to, you know, to look for the organisms. And so in the oral cavity, you know, being able to look for material in there and to look for bacteria kind of falls in the category of looking, you know, at tissues. So we can be looking for, you know, for the tissue in the mucosa, is it on the cheeks, is it on the gum line, underneath, you know, where are we trying to get this material from, you know, tattered calculus, where are we finding this stuff? And so we need to be able to scrape, collect, and again, we're putting things in place, we're putting things in liquid media, right? You know, we're making, you know, suspensions of them, you know, to look under the microscope, we're staining with gram stain, all of those things that we talked about yesterday. And so, you know, we can analyze what we're collecting, you know, by growing our media if we can, everything to, if we cannot, you know, doing molecular methods like PCR and, you know, sequencing, you know, the products. Serology is pretty big, you know, looking for antibodies against, you know, antigens of these organisms, right, using a variety of immunoassays, you know, everything from ELISAs to Western blots to immunoprecipitations to IFAs and so on. So, you know, microscopy, pretty standard, you want to be able to see right away. And as we will talk about, you know, later on, you know, everything, you know, microscopy is everything from just a regular compound microscope that you would have available, all the way, you know, to confocal microscopy, all the way to atomic force microscopy, you know, to electron microscopy, right? So depending on what's going on, multiple methods of, you know, brought to bear to understand what we're looking at, okay? So we want to kind of remember those things. And so again, we're seeing examples of those. So, we can do PCR, we get the products, we run them out in agarose gels, we can see the bands, we can take, the products, we can have them sequenced, and we can blast, the sequences, right, to find out, what they match in the databases. And so what you see up there is just a typical agarose gel, okay, with ethylium bromide stained DNA, okay, which is why you see the bands, the fluoresce, you can see the white bands. And so we can do fluorescent in situ hybridization, also molecular methods where you actually prepare probes, okay, molecular probes, and you label them with the fluorescent tags, and then you use those to hybridize the DNA, that you've now, prepared from whatever source you have, and you will see wherever you have interaction and hybridization, you will see the fluorescence, okay? And so again, we're just reminding ourselves, the different phyla that we talked about, so we can see the representation again, right? So we have the actinobacteria, Firmicutes, Proteobacteria, and the Bacteroides. So we all remember those 4 major ones that we mentioned yesterday, okay? And so we need to remember this schema right here. So, you'll have, questions, just kind of making sure that we know the flow, right? And so, we can obtain the sample, from the teeth, we can get a DNA, we can do the amplification, by PCR, right? We can actually, ligate the product into vectors like a plasmid, we could do that. We can transform E. coli, and we can actually, screen to make sure we have the inserts in there, okay? And then we can also sequence that DNA. And so we can generate, profiles that can let us know, right, specific organisms as well as, within genera, okay, or within groups or even within the phylum, okay? So, but, we need to kind of remember the scheme right here, beginning from, from the teeth over there, okay, all the way to getting the sequence information, and then being able to get a sense of, what organisms the DNA matches, okay? So, again, just look at the pattern. So again, if we're going to ligate, even though it's not mentioned in there, right, we're going to treat the DNA with restriction enzymes, right, so that before we can, get the hybridization of the vector, and then, with the ligase, make sure that we have the fragments in there. Somebody's hand was up here.
Yeah, is there a name for this specific technique or just DNA sequencing?
Right, so basically, this is a cloning technique, okay? Once you're into the vector over there, so essentially how we detect the DNA. So, a broad name for the procedure would be, detection of DNA, bacterial DNA from, samples in the oral cavity, into the teeth, okay? So, it makes sense? So again, if we're going to ligate in there, I just want to make sure we get that point, even though it's not on the slide, that we're going to, restrict the, the DNA that we've isolated and the vector that we're going to put it in, okay, before we add the ligase, the enzyme, that's going to allow, the recombinant to be formed, okay? So once you have this right here, once you have the vector plus the piece of the DNA that's been amplified, this thing over here is called the recombinant, okay? And then this is what's now put into the bacteria, okay? E. coli is a common host cell, for doing this, okay? And so the sequence information right here, right, is when we keep saying that we're going to search by blast, okay? This sequence is what's now put into the blast search to do the matches, okay? Any questions about that schema? Okay. All right,
Yeah.
go ahead.
What was the purpose of treating the DNA with restriction enzymes?
Right, so you want to cut the DNA. So you want to cut the plasmid DNA open. So the plasmid is typically circular. So you want to cut the DNA on the plasmid so that you can insert the piece of DNA that you've amplified. Right. So in order for you to do that, you're going to treat both the plasmid and the insert with the same restriction enzyme so that you generate the same sites. And then you put them together, and then you ligate with the enzyme. Okay? Any questions about that still? Everybody kind of clear what's going on? Okay. All right, so, this table, again, just kind of, gives a brief, comparison of the bacteria and archaea, the features. So again, remember this, they're both, prokaryotes, and, but two domains. And that's because we do have some differences between the archaea and the bacteria. But some of the general things that we've talked about, are pretty similar. So the 70S ribosomal size, for example, the plasmids are found in both. We can have some linear plasmids that we have in bacteria. Other things, that are common, to them. If we look inside, we can see inclusion bodies. We can see them in both types of cells. The cell wall component is very different. So we have the peptidoglycan, right, that tells us we're looking at bacteria. So remember, right before we left yesterday, we kind of drew the little flow chart, right? So we have the prokaryotes, they have regular peptidoglycan, bacteria. If we have, this kind of different, cell wall, pseudomurine type thing, then we're dealing with the archaea. Not found on this table right here, but, the archaea are typically associated with extreme environments, okay, for their growth. And, the regular, if you will, bacteria are found in more typical environments and so on. And so all the way down to the, to the last feature over here, where when we talk about bacteria, we can talk about them possessing capsules and so on. And that's, common feature, you can see them in gram-positive, gram-negative cells. With the archaea, we don't typically have that. They will have features that allow them to adhere to substrates, right? So we can see that in both archaea, we can see that also in regular bacteria as well. So we have a plasma membrane in both cell types, but we have different types of phospholipids, and so on that we have associated, with each cell membrane. And so again, that's just something to allow us. So we want to look at the cell shapes and arrangements. So we're going to need to remember this just, for example. And so we have, the first type of arrangement over here, the cocci or spheres. And we can have, depending on how the cells divide and what plane they divide, we can have, the cells, in pairs. We can have them in chains. We can have them in clusters. And so we have diplococci, streptococci, staphylococci, again, letting us know, what kind of arrangement they have. We can have them in tetrads. We can have, assassinae. We can have, this 8 cells, it's kind of this cube arrangement. And so, sometimes, when I go through this kind of material and with other classes, and it's like, it's so tedious, why do we have to know all these different shapes, and, but we do, because, we use them to kind of differentiate the different organisms. So Staphylococcus, aureus, when we talk about, the grape-like clusters, as soon as you say that, you have something in mind, okay? And so it's important, to know these structures. So we have other kinds of arrangements. We have the rod shapes. We can have cocobacillary type organisms. They're kind of roundish, kind of rodish looking. We have the comma-shaped organisms, the vibrios. We have, spiral-shaped, things. And the spirilla and spirochetes are not the same. So yesterday we had, on one of the lists, we have Treponema on the top, and we said, Treponema is usually found and sometimes with the Porphyromonas gingivalis, right? And so again, there we have this, organism, that's spiral-shaped, right? So when we see that, we're thinking of something like a Treponema. So the spirochetes and the spirillum, okay, have to do with flexibility and rigidity of the helix that's formed. And also, the movement, of these spiral-shaped, organisms, right? So again, it's important to know. So we're seeing examples, over here. And so, again, you see, spirochete, you see a spirillum in the middle, right? And we see the vibrio, comma-shaped, that we have. And so things like bacillus, we're always thinking of these rods and chains. They're pretty common. And we have other shapes and so on. And so we have things that, form these filamentous, type things. They almost look like they're fungi, but they're not. So we have mycelium, we have things that are pleomorphic, so that, within, if you look at a smear, you're going to see variable, shapes, of the same organism, okay? And we have, let's see. Okay, so we have others, that have found within the bacteria. So we have things that have, stalks and prosthesis, if you will, on them. So those we don't encounter very common, in the body and so on. But again, they have, different types of shapes as well. And so we have a nucleoid in the bacteria, right? So we have, the DNA, the cytoplasm, and typically we have the circular structure. And so this is what, we're seeing in there. So we can have, situation where we have associations with proteins, but we don't have histones like we have in eukaryotic DNA. So that's another thing that's used to kind of distinguish, these things. And so bacterial DNA doesn't have that. So the plasmids, we have, the exosomal DNA, they're typically circular, right? They're double-stranded DNA molecules. They can replicate, outside of the bacterial chromosome. And, they can be very important for pathogenic states. So that plasmids can contain the genes that encode proteins that impart virulence to the bacterial hosts, okay? So they can do this, by having encoded proteins, enzymes that can destroy antibiotics, for example. They can encode proteins, in many cases also enzymes that allow the bacteria to be invasive. And they can also encode toxins, okay, that allow the presence of that bacteria, right, to be highly pathogenic within the host as a result of the toxin that's produced, right? So the plasmids, can be very important. And as well, the bacteriophage that infects, some of these bacteria can have the same function. All right, and so they're used for classification, right? In terms of the type of organisms, that have them, the particular genes that are present, the products that are encoded by those genes, and so on. And we're going to see that within biofilms, right, this is very important because we can exchange, the plasmids, and as well, we can have a phage that can affect, the host in there. So on the table over here, the two items that are circled, we want to remember those. And so again, we're looking at, plasmids that carry antibiotic resistance genes, and we have, ones that carry virulence. So those two features of plasmids we want to remember, okay? The ability to confer antibiotic resistance and the ability to confer virulence to the host bacteria. We want to make sure that we remember those two properties, right? So there are other things that, the plasmids can do, right? So, yeah, but the two, the circles, we definitely want to remember those two. All right, so structures of the bacteria. So we want to be able to, so if you see a list of these, so many times the exams just want to make sure that, we definitely know characteristics and features of bacteria. So, they'll list a number of things and ask you, these eukaryotic features or other bacterial features and the prokaryotic features. And so, yesterday we looked at a number of things. We said flagella, we can have on both prokaryotes and eukaryotes. The way the flagella, is structured in the cell is different from bacteria to eukaryotes, okay? But just having flagella there is not a unique feature of bacteria. Does that make sense? Okay? All right, so capsules, we have on bacteria. Formation of biofilms, we can have, bacteria will do this. The presence of plasmids, having, fimbriae, having pili, these things, we associate with bacteria. We can have specialized structures for the cells, depending on what the cells do or where they're found. So if they're photosynthetic, thylakoids, gas vesicles, things that are in aquatic environments. A whole variety of storage granules, sulfur granules, we have those that have magnetosomes in them. So all these are things that we associate with bacteria. So we want to look at the different features of the bacteria in a little bit more detail. So in the cytoplasm, what do we have in there? So we already mentioned the DNA. We also have, the various, RNAs in there, proteins. We also have inclusion bodies, okay, as we just saw in the list before, that can be found in the cytoplasm of the bacteria. So we mentioned yesterday that we do not have an endomembrane system inside bacteria. Does everybody kind of remember that? Right? So we don't have Golgi, we don't have, ER, we don't have lysosomes, those kind of things, right? Those are eukaryotic cell features. We have, cytoskeleton, in the cells. And so just like we have cytoskeletal proteins in eukaryotic cells, and we can talk about, actin and tubulin and spectrin and all these different type of things, people have been very interested in cytoskeletal proteins in bacteria to see if there were equivalents in bacteria, just like we have, in eukaryotic systems. And so the cytoskeleton is important because it kind of maintains structure, plus within the cell, can serve as tracks for the movement of vesicles. Okay, so you kind of remember this a little bit. So we have all of those things. So people are also interested in bacteria to look at things. So we have, proteins that, found and function in, in the synthesis, of the cell wall, and when the cells divide. And so a lot of that work has now, shown that, the FTSZ protein is essentially equivalent, to the tubulin that we have, the MRB to actin, and then so we have, other proteins that are equivalent to intermediate filaments in the cells. And pretty much what the images show you on this side is the role of those proteins, in cell division and in cell wall synthesis. So again, just like we have in eukaryotic cells where we have the function of actin and tubulin, we see similarities in here. And I added a couple of pictures that you don't have in your notes, just kind of highlight this point over here. And so we see other proteins similar to the intermediate filaments, that we have in eukaryotic cells that are important for moving things. So we all remember that when we move vesicles within the cytoplasm of eukaryotic cells, right, we have them on tracks, filaments, as they're making their way up towards being in the membrane for exocytosis, for example. So we have similar kind of things inside a bacteria. And so that's what we're seeing over here. So again, we have other proteins that are important, right, for moving, the plasmids around when the plasmids, undergo replication and things like this. And so I added, these next two slides, just to highlight that. So again, the proteins, that the main ones that we're mentioning, the FTSZ, okay, we have there, it's a tubulin homolog, so we need to remember that. The other one we mentioned, MREB, MBL, proteins and actin homolog. And then, we have, the presenting protein that we have in Colobacter, one of those stalked bacteria. And so we have, homologs of intermediate filaments. So those three proteins we need to remember, okay? So, we have the FTSZ, we have the MREB, MBL, and then we have, the CREST, protein. So those three we need to remember. So there are other ones in there, as you see, right? So we have, just like we have in the cytoskeleton of the eukaryotic cells, we have a lot of different proteins. We can talk about ankyrin and spectrin and actin and, all of this. We have a similar kind of situation, okay? So, but the three that, we're wanting to kind of really highlight on are the three that we've mentioned. So we want to know those. All right, so here they are again, okay? And again, we're focusing on them because these are the best studied examples of them, okay? So what I will do is I will, I have, the 3 slides, 2 of these we just seen and one where I added some information that I will send, okay? I'll send those so that you can have them, okay? So again, 3 best studied examples, okay? So knowing what they do, so forming, that Z ring, okay, during the division, and then maintaining the shape, getting that peptidoglycan, as we're going to see, having it moved out to the, to the surface of the cell, and then, maintaining shape, in particular these stalk bacteria, this particular protein is very important in maintaining shape over there. So these three, we want to make sure that we know. And, there are ones that, amongst all the different proteins we had on the table, are ones that, typically you will have, questions on, in other exams, because those are the ones that you have a lot of people really focused on, right, trying to study to understand, how they could be targeted, okay, for therapy and so on. Okay, so these are pretty important. Okay, so we have the cell envelope is the cell membrane, plasma membrane. And so again, we have, a typical bilayered membrane with the phospholipids. And so, this kind of maintains the cell integrity, okay, so it separates the internal portion from the outside. And so we have the phospholipids in there, we have, proteins embedded in the membrane, we have proteins that are peripheral, ones that are integral, so we have all these properties in there. And so what you're seeing up here is, the depiction of the phospholipids, with the fatty acid tails. And this is the, essentially, the cartoon, where if we see this, we know we're looking at a phospholipid, the circle with the two tails, right? And that's what we have in here. So we're looking at that bilayered membrane. We have integral membrane proteins, we have peripheral membrane proteins. The interior is hydrophobic, right? And then we have the lipid tails, posed in there. And then we have the charged heads facing, the external surface and cytoplasmic surface, okay? And so we need to remember that orientation. We can have, in the membrane, cholesterol, and in bacteria we can find some, similar type of steroid type molecules, the hopanoids, ones that have been described, in bacteria. And so, again, depending on where the cells are found, what environments they can tolerate, kind of dictates, what kind of these steroids you find in the plasma membrane. So the key thing we just need to remember is that the bilayered membrane, so all cells have plasma membrane. The bilayered membrane with the features that we just talked about and the orientation of the phospholipids. And so the glycolipids, will have the sugar moieties facing the outside. Glycoproteins in those membranes will have the sugar moieties facing the outside. So we always look for the carbohydrate molecules on the outside, on the surface of the cells. Okay, and so things will go in and out of the membrane, either coming in, going out. Excuse me. We have movement of gases, across the membranes. We can have, water, small amounts. We have large amounts. Then we have aquaporins, water channels to get, water in. And so osmosis, will occur across the membrane. And typically, the water flow follows the solute concentration. And so, again, we can have the cells swell, or if the water goes in, or we can have them shrivel if they lose the water, right? And the fact that the bacteria have the cell wall kind of helps the cells, deal with changes in salt concentration in the environment. And so nutrients are also taken in. Okay, we can have them diffused. They can come through channels. So they can do so without energy use. We can also have active transport take place with the use of ATP hydrolysis, and we can have things come in, binding to receptors getting in, or coming in straight through the channels. And so we can have co-transport mechanisms, and we can have group translocation of molecules, and so on. So so these are the things that I added on here on the top, okay, of your notes. So this is that third slide, so I will send those 3 slides. And so the whole point of it is to have us remember the differences, so we can have diffusion, passive diffusion, we can have active transport, and so we just want to realize, the differences that we have, And so if we do active transport, we can do primary active transport, so ABC cassette type molecules in the membrane will allow solutes that are being brought into the cell, okay, carried on the binding proteins. They will bind to the receptor, change of conformation occurs, right? The solute is now transported in. We have ATP binding sites, on the receptor. We hydrolyze the ATP. We can open up the channel, get the solutes in, and then conformation changes again, awaiting the next transport. Does that make sense? And so that's the primary active transport we have in that case. And the example of that is a uniform. And so again, we just have stuff, just going through in one direction. We can then have also secondary active transport where you have ion gradients that are used to couple the transport. And so we can see, examples of that over here. We can have either symport, both things being transported going in the same direction, or, things being transported going in opposite directions. And so bacteria will do this. And we can also have modification of what's being transported. For example, they can become phosphorylated, okay? And then, we can now move them, or once they get across, we phosphorylate them and they stay inside. Okay, so these are all ways in which we can have bacteria transport molecules. So we'll look at these next couple of slides, and we'll take a little break here. And so here's the cell envelope. Okay, here's the example of the ABC transporter. So we're bringing in substances. Here we have the sites where we're going to have the ATP bind. And so again, we bind to the binding protein. It interacts with the transporter. We change the conformation, we can have ATP bind, okay, we open up the site, we bring in the solute. And then we change conformation again so that we can do the binding all over again and transport. And so, in all these different things that we've talked about, okay, so when we look at bacteria and environments, and so our focus beginning next week, Tuesday, will be, on Monday, will be in the oral cavity. So when you have bacteria in these biofilms, right, they're metabolizing, we already talked about the plasmid changes, being moved around. We have changes, within the environment of pH, salt concentrations, all the things that will allow the biofilm to survive. And so one of the difficulties with biofilms is that the organisms that you find on the periphery, as we're going to see, right, if you're trying to have antimicrobial agents against them, you can interact with those on the peripheries. The ones in the middle are the most difficult ones to get to. And by design, the biofilm is organized in such a way, okay, as we're going to find out. And so, nutritional requirements, pH, oxidative, whether you have aerobic and then going into the core, more anaerobic, gene exchanges, all of these things contribute to the virulence and disease-causing ability of the organisms, okay? So we kind of want to keep that in mind. So let's take a break, 5 minutes or so, and then we'll continue. Any questions? Okay. So let's just take a little break. So again, so we're still looking at that cell envelope or plasma membrane. And so what we also want to remember, so if you're, thinking of bacteria and metabolism, and if we have oxidative phosphorylation taking place, right, so the electron transport chain, all of those different things, associated with plasma membrane, so we want to kind of remember that. And so, we have a number of important aspects, not just, for maintaining cellular integrity, but really, important functional aspects of that membrane, of which, one of these you see over here for energy production. And so we generate, proton motive force, we're on that membrane, we're doing oxidative phosphorylation there. We want to be able to have that energy available, for the cells have flagella, that's where they're getting the energy from, to move that flagella, and so on. So a lot of things that will happen to bacteria in the environment, okay? They're sensing, the changes in the environment, different gradients, whether it's everything from nutrients all the way to toxic, components those will be sensed, again, using that membrane. And so we want to look a little bit closer at the cells, okay, so gram-positive, gram-negative, and so we want to remember these parts of the cells. So I have, two sets, one using the illustration from one of the texts, that was assigned to the class. I'm not sure if in the last year or so if it's still on the list of books, but in any case, so this figure is from there, and the next one, and then I have, other figures and more of, pictures that show the same thing. And so we're looking at gram-negative cell over here, looking at the cell wall. And so we have, the plasma membrane, okay, or cell membrane, we have the periplasmic space, and we have that thin layer of peptidoglycan. We have, different types of lipoproteins, and then we have that outer membrane, okay? So the outer membrane is, outside the peptidoglycan layer, okay, of the bacterial cells. And so we have the lipopolysaccharide, or LPS, embedded into that outer membrane. And so depending on the type of gram-negative cell that it is, we have different porins and transporters and channels that will be found in that outer membrane. Okay? So again, the LPS is a major feature of these gram-negative cells. And so we mentioned some examples of the gram-negative cells yesterday, ones that we're familiar with. We mentioned Ecology, we mentioned Salmonella, we mentioned Shigella, all the various enterobacteria, all gram-negative organisms. So they would, kind of be organized like this. And because E. coli is not, Salmonella, right, you're going to have features that will distinguish, Salmonella species from Escherichia coli, right, like that. So this is gram-negative. And then we have the gram-positive cell wall, okay? And so we have the plasma membrane, okay? We have the cell wall, so you can already appreciate how thick the peptidoglycan layer is. And then associated with that peptidoglycan layer, right, we have a variety of structures. So we have lipoteichoic acids, okay, with a lipidic component to them. We have the teichoic acid by itself, more water-soluble, substance, and these we have, in the peptidoglycan layer. We have, different surface proteins that can be also found, associated with the peptidoglycan, and those lipoteichoic acids, we can have, embedded into the plasma membrane, right, in the top leaflet. So again, already, so here's the gram-negative cell wall. Right, and here is the gram-positive cell wall. I mentioned to you yesterday, and you'll see this, depending on what text you're looking at or what article you're reading, right? There's a little, small periplasmic space, okay, on the gram-positive cells. It's very tiny, so it's not usually highlighted, okay. Here it's highlighted because it is quite a big space, about 70 to 100 nanometers, huge space. So inside there we have a lot of substances that are put in there by the cells for transport. You will find a variety of enzymes in here. Important ones, to mention in the periplasmic space, here, and also in the space, for the gram-positive cell, is enzymes that can, destroy antibiotics. So we have, the beta-lactamases that are present that can go after the beta-lactam ring that we have on things like penicillin. Cephalosporin, so important stuff in there. And so, again, we want to know the differences between the two, okay, so we see them. And so, we also have, again, this little schema over here is just to kind of make sure that we are complete in mentioning it. So we can see, the movement of molecules, in and out of that membrane. Okay, with the gram-negative cells, when they move out, they'll get into the periplasmic space first, and then, they'll move on towards the outer membrane, right, out to the environment, okay? So in the gram-positive cells, we don't have that outer membrane. So when we get things out, right, they're coming out to the surface. And so, again, the transport mechanisms we talked about, so we can either do uniport, symport, antiport, all of those things. Diffusion, facilitated diffusion, active transport, all of those things can take place across the membrane. So we want to look a little bit at the cell wall of the bacteria. And so the cell wall is pretty important for these cells. Now we do have organisms like mycoplasma, chlamydia, that don't have the cell wall that we were talking about. And so when you see cell wall-less organisms, those are usually mentioned. And so, the typical, bacteria, okay, except for the things like the gram-variable ones like mycobacterium and so on, that are acid-fast, again, their cell wall has different composition. You have mycolic acids in there, very waxy, lipidic stuff. And so it makes it really difficult to do the gram staining with those cells, okay? So the cell walls we're looking at over here, and again, the general cell wall organization of getting that peptidoglycan made and transported, across the membrane to the outside. And so we have, the general structure of these, the peptidoglycan molecule. It consists of, alternating molecules of N-acetylmuramic acid and acetylglucosamine. So we have, the two molecules, right, alternating in this, chain of a molecule that encases the bacteria, okay? And so we have this, thing is cross-linked, it's put together, and then it's transported, to the outside. So again, we want to remember the NAM, N-A-M, okay, the N-acetylmuramic acid, the NAG, the N-acetylglucosamine. So these two things, okay, these two molecules, what we see overlapping. So generally, we have the unit look as follows. So, we have both the NAG and the NAM, okay, they're associated. And then we have these peptide chains that are linked to the unit. And so we can have, things like alanine, lysine, glutamate, they're part of that. And you're going to see the differences based on cell type, okay? And as well, when we look at how things are cross-linked, we're going to see differences between how gram-negative cells would do it versus how gram-positive cells would do it. Regardless of, of whether we're looking at gram-positive or gram-negative cells, okay, we already saw in the picture, both of the cell types have the peptidoglycan. The unit is very thin in the gram-negative cells, but it's there, okay? And in the gram-positive cells, it's pretty thick. And this has implications for, when we use antibiotics that are directed against, the bacteria, we find some that are pretty broad, okay, that can attack, both cell types. And the difficulty in, getting at that peptidoglycan is that outer membrane that's there, okay, in the gram-negative cells. So again, we have the N-acetylmuramic acid, we have the N-acetylglucosamine, right? And then we have the peptide that we have associated with them. And again, it's usually associated with the N-acetylmuramic acid. And so we see, the way in which things occur. So we have, the alternating NAM-NAG. And then, we have the peptide associated, with the NAM. So again, tetrapeptide. And then we have that, one unit linked to a second unit, okay? So we can have a direct association of the alanine to the diaminobutyric acid, the DAP, okay, in the gram-negative cells. We can have a direct cross-link. Or we can have the example as we see in, in some of the gram-positive cells, where we have, a pentapeptide, okay, of glycine that links one unit of the NAM-NAG, okay, to another, okay? So we need to kind of remember this, okay, the differences that we can see in gram-negative, gram-positive. So what we want to remember, this core, okay? So we have this alternating core that we have linked, okay, and that's what we're seeing over here. And so we can have a direct link in all the gram-negative cells. We can have this, this pentaglycine. And again, depending on the gram-positive cell, we can have a mixture in here, okay, amongst the glycines. We might see like an alanine or something, right? So, we can see differences. And again, it depends on the particular organism, okay? But, so this is a general co-arrangement. And so we see the use of D amino acids, right, which we don't typically see in the proteins that we have in cells. So L amino acids are what we usually see. But in the cell wall, we have this here. And some of the thinking behind this is that this prevents the peptidoglycan from being targeted by proteases easily, okay? So it helps, to maintain the integrity of that peptidoglycan. So this is a little bit more detailed information again from that same text, just kind of showing you the way in which, the peptidoglycan is synthesized. And so, we start out, we have, UTP process, right? We add the amino acids, to the sugar, and then, we begin to go through the process of cross-linking, right? And, we cross-link, have the whole tetrapeptide associated with it, and then now we have to attach the glycine units, the 5 glycines, if we're looking at a gram-positive cell, and then we have to translocate that entire thing across the membrane. And then once we do the translocation, then, we do the cross-linking outside, and then we firm up the cage, okay, around the bacteria. So, it's important, to remember this, but, for our purposes in here, what we want to remember is the unit. So when we have the NAMNAG, alteration, right, the tetrapeptide associated, and then knowing that we cross-link that to another unit. And so this repeats over and over again to form the whole cage around the cells, okay? And so if any of these processes are targeted, so if we can't get the tetrapeptide core, if we cannot link the NAM to the NAG, if we cannot translocate, right, then we can't have a fully functional peptidoglycan on the cell, okay? And so this is the reason why, in the clinic, both medically, dental, etc., we name it, these cell walls are huge targets, okay? And so because the bacteria can produce the enzymes that can disrupt, the antibiotics, we have antibiotics, that are done in combination now, so that if we destroy the beta-lactam ring, we still have another functional unit of the antibiotic, that can still help to kill, okay? So, a very active area of research is the translocation. How do we get that thing across the membrane and, form the cross-links on the outside? That's a big area of research. Another area that's important is how do we do that, cross-link, we'll put that bridge, okay? And why do we have in some cases, the 5 glycines or a mixture of glycines and alanines and all of that. And so, there's a lot of information in the literature regarding what, people are doing with this. And so again, we just wanted to get another look of the same thing, a little more simplified, right? So again, we synthesize, those 2 sugars, right? N-acetyluramic acid, N-acetylglucosamine, right? We make them, so just in the typical way you think of your monomer sugars, being linked and so on. And then, in this example given, okay, we have, the bactoprenol that can be linked to that as well. And so the idea is now to be able to get, the unit across the membrane, and then, to have the peptides cross-linked, so that you can now have this cage outside. Now, there are other molecules, besides bactoprenol. There's a lot of interest again, in 2 questions. One, okay, how do we get this huge thing across that membrane, okay? And when we get to the outside of the membrane, how do we orient it so that the cross-links, will occur in the manner in which they look like when you isolate them from the outside? So those are, areas that are getting a lot of interest. And so what this bactoprenol is supposed to do is to be able to, kind of get the thing across, flip it, and get it the right orientation. But then the question still remains, how? Are there other molecules that work with it to do so, and so on? So let's, you see different variations of what people are finding for that. So we want to know that. So again, we look at it again. right? So we're seeing, you know, the 2 sugar units, we see the tetrapeptide, and what this is showing is that enzymes, you know, like lysozyme can cleave. Okay, so you know, part of our innate immune responses is to have, you know, lysozyme that we can use to destroy bacteria. The bacteria can cleave, you know, the peptidoglycan backbone, and when that happens, you know, the cells are vulnerable and they can die that way. We can also, you know, have, you know, the lysostatin also, you know, do the same thing, right? We can disrupt that pentapeptide, and again, we can destroy the cross-link. And we can also have, you know, the antibiotics, you know, that we, again, you know, that we use. And again, these can disrupt that backbone. And so again, when that happens, you know, the peptidoglycan structure, you know, is disabled. And so we can also have other things, you know, that can affect the cell wall. So in when we have the cell wall, you know, disrupted, if we have gram-positive cells, you know, they become what are referred to as protoplasts. And so I highlighted, you know, with the red, you know, just to make sure that, you know, we take note of that. The same thing happens to gram-negative cells, you know, they become what are referred to as spheroplasts. And again, this goes back to remembering what we just said about them. So which of the two has the thicker peptidoglycan layer? The gram-positives, right? Gram-negatives have the thin, you know, peptidoglycan. What do they have outside of that peptidoglycan? So, right, so before we get to the LPS, we have the outer membrane, right? So on that outer membrane, right, we have, you know, proteins embedded in it, including the LPS, which is there, right? And so when you strip that off, you know, you're going to have the cells kind of look a little different, right? So the protoplasts, okay, you know, the gram-negative cells, the spheroplasts, you know, the protoplast gram-positive, spheroplast gram-negative, when the cell wall is, you know, disrupted, okay? And so these cells will have problems with, you know, dealing with osmotic pressure and so on, because now that cell wall material, you know, is no longer there, okay? So we need to remember those two terms, okay? So again, we're seeing what can happen, right? We disrupt the, you know, the peptidoglycan. We're looking at, you know, a rod-shaped organism over there. Okay, so again, you know, we have the active condition, we have it over there with the protoplast. Okay, if we have it, you know, have other conditions, we can break this, you know, the cell, and we can have stuff, you know, leak out of the cell. Okay, so we can also, you know, again, we talked about the fact that in that periplasmic space, you know, the cells can put enzymes in there, you know, with other proteins, and one important one is the beta-lactamase. And so, you know, penicillinase, okay, is an example of a beta-lactamase. And so it can break the beta-lactam ring, okay? And so when that happens, you know, that essentially, you know, the ability of that drug to work is, you know, is no longer effective. But then, you know, we have these, you know, pseudo-synthetic type things where you can add another ring, okay, add another component. And so if we break the ring, we're still effective, and so on. And so, you know, this is an example that we have with something like amoxicillin. So again, you know, that periplasmic space, you know, pretty important for these organisms. Okay, so this next set of slides, you know, just to kind of reinforce the things we've talked about. So again, please remember the proteins that we have, you know, the molecules like this. If you see lipoteichoic acid, right? So you know you're looking at gram-positive cells. If we see LPS, we know we're looking at gram-negative cells, right? If we see the mention of lipid A, we know this is, you know, component of LPS. We're looking at gram-negative cells, and so on. So these additional, you know, pictures just kind of give us the same, just, you know, another look at the same structures, just so that we remember. And again, periplasmic space in the gram-positive cells is very small, okay, compared to the, you know, big periplasmic space that we have for gram-negative cells. And so again, you know, the electron micrograph, you know, over there on the left, you know, kind of shows you, you know, the same things that we're showing by illustration. Basically letting you know that we can see those things if we prepare the cells, you know, by microscopy, okay? So, again, gram-negative cells, okay, we see that, big periplasmic space that's there. We see the outer membrane, and then we have the LPS associated with that. So, so everybody please notice structure, okay? So notice organization of the LPS molecule, okay, with the O side chain, okay, the O antigen, the core polysaccharide, and really when you look at this, there's really an internal and external portion of the core. So the external being the part nearest the O antigen, the internal being the part nearest the lipid A, and then we have that lipid A, okay, and then we have those inserted in that outer membrane, okay. So again, when we see this, we know we're looking at gram-negative cell component, and the importance of LPS, as we had the question before, and this is significant even in the organisms we have in the oral cavity, is the shedding of this LPS. So when the organisms die, the LPS releases, and so we have a whole host of activities that are directed towards the LPS. Everything from having toxicity, we have septic situations where it naturally lead to death. We mentioned the myelogenic situation, with that. Inflammation, and so many of the organisms that we are used to thinking about, during inflammation, so, things like tumor necrosis factor, inflammatory mediators like prostaglandin. We also mentioned the B cells and the myelogenic activity, which is specific to B cells. So B lymphocytes, will begin to respond. Complement activation, leukocyte activation. The LPS can behave like an adjuvant, and so on. So we can have a whole lot of responses to that molecule. And in the molecule, the most important part, okay, for these activities is the lipid A component, okay? So the O side chain or O antigen is important for other reasons. So the immune system can target the O antigen, and we can have antibodies that are made and so on. And so those antibodies are used in serological typing of the cells, right? So you can have variants of given organism, right, with the changes that occur in that O side chain. And so the antibodies that are produced can be used for typing. Okay, so you can identify different strains of E. coli. So when you see the designation of the strains of E. coli, and you see that O, everybody's seen that, it refers to that O side chain. Does that make sense? Okay, so that's used to designate the different strains. Okay, so please remember this this structure. The lipid A part, the core polysaccharide, and the O side chain, and the importance of that O side chain for serological typing. So, we mentioned the business of having things go in and out of the cells, and so again, see that, outer membrane, we have the porins, we have transfer proteins, an example of them, the PUN-B, and so we can get things coming in to go into the periplasmic space and then go into the cell. And as well, in the other direction, we can have things coming across the plasma membrane, into periplasmic space, and then out. And so, because that space has all those molecules in it, right? So, people who study, the delivery of molecules into cells, nanoparticles, that they can charge and get into the bacteria, whatever, they focus on that space. So if you can get stuff in there, then you can get it into the cell, okay? And so, because that peptidoglycan is so far inside, because of the outer membrane, you want to be able to exploit that space to get things into the cell, and so on. So, all of the different things that we mentioned, the plasma membrane doing, that will occur, but we don't want to forget that we have that extra membrane, that outer membrane on the outside, okay? And so again, pretty much, what we're seeing over here, different ways that people think about, how to get things in and out, okay? And so we have, these auto transporters, we have this single-step transporter units, so because there are all these different, attractive ways, if you will, people really want to see how can that be exploited for the delivery of molecules that will be detrimental to the bacteria, okay, when you're doing treatments and things of that nature. Okay, so we have some questions over here, just kind of help us remember the things we've been talking about. So which of the following structures is not associated with gram-negative bacteria? Okay, so which one?
C.
C. Okay, so everybody remembers teichoic acid. What's the other molecule? Lipoic acid, right? And we have, the gram-positive cells, right? Okay, and so yesterday we had this one, and sometimes I have a picture, but okay, so if you do a gram stain, right, gram-positive cells will stain, okay, and then gram-negative cells will stain red, okay. And so everybody kind of makes sense? Okay. And so we just looked at the peptidoglycan subunit composition. Okay, so which one is it? Okay, I hear A and I hear something else that's not coming out very loud. Which one is it? A. Everybody in agreement? Okay, so we want to remember that, okay? And again, that variation, remember, we can have either direct link, okay, or we can have the pentapeptide. Okay, so gram-negative cells, typically you're going to see that, direct link. Gram-positive cells, you're going to see, some variation of either all 5 glycines or glycines mixed with alanines and things like that, okay? And then of the tetrapeptide coming down, also you're going to see a mixture of alanines, lysine, glutamate, okay, and so on, that's part of that tetrapeptide, okay? And then we have the 2 sugars, right, the NAM and the NAD, okay? So we remember those. All right, and so this table kind of summarizes things, to look at, and so we just kind of want to, remember down here with a capsule, okay, that we can have, that is really important. It's an immune evasion structure for bacteria because it makes it difficult for the macrophages to do the phagocytosis. The other molecule that are down here, the S layer or surface array layer, is a protein layer. And this one, I mentioned the nanoparticles, just a little bit ago. There's a lot of interest in that S layer, also for therapy. And one of the reasons for it is that the S layer can self-assemble, okay, in these, plates, okay. And so the idea is that, again, if you can do the self-assembly, maybe you can incorporate, a toxin or something, antimicrobial agent in that array that, once in place can affect the bacteria and you can kill them that way. So people who do, nanoparticle type work, nanotechnology are very interested in the self-assembly of the S layer. Okay, and we have the S layer in both gram-positive and gram-negative cells. Okay, and so that's a really important feature that people like. And so use this table to kind of help you kind of summarize, the things that we've been talking about, that differentiate the cells. So we're not going to belabor these. We just want to mention, the function of some of these structures. So again, we know we can have flagella, okay, for motility and so on. And we have different, aspects of motility depending on whether there's one flagellum, okay, or whether you have, a flagellum on either side of the cell, or whether you have the whole cell covered with, with flagella. It's going to make a difference, in how they move. And because you have these differences, they're actually important for characterization of the organisms, okay? And so we're seeing, an example of a peritricious situation with a flagella, but sometimes we can have them be more numerous all around the cell. I have these cultures that I'm working with right now that have a lot of bacteria in there, and it's always interesting when you have them under the microscope, to actually see just, in the tissue culture, just having the flask under the inverted microscope and just seeing all this bacteria kind of moving around, that have different, types of flagella on them and so on. And it's always really interesting. You have ones that are very rapid movement, within the culture, and others, like the spiral type ones that are a little bit, different, the way they kind of twirl their way around, the media. So very interesting stuff to look at. And so we have the flagella. The only reason we're looking at this is just to say a couple of points. that the organization of the flagella in bacteria is different from that in eukaryotic cells. So in eukaryotic cells, we have the microtubular arrangement, the 9+2, okay, the microtubules, right, that we have in cilia and so on. But that arrangement is not the same here. Okay, so here we have this, this kind of machine that's put together, and you have, the hook portion, the filament portion, the body, the basal body into the membrane that you have now stuck, in that membrane. And so it's a whole different arrangement from what we see with flagella in eukaryotic cells. And so, that's the thing, we just wanted to make sure we mentioned that. And so we have, cells, that have the flagella organization, within the cells that also allows for motility. So, which means, not all the cells that have flagella have them sticking out on the outside. And so, in the organisms we're going to encounter in the oral cavity, we're going to be paying attention to, which ones, of course, are gram-positive, gram-negative, which ones are rods, which ones are cocci, which ones are cocobacillary, okay, gram-positive, gram-negative, but also additional features that we're going to be looking at, types of adherence molecules that they're producing, any particular enzymes of importance that they're producing, right, that help them to establish the biofilms, okay? And so, it becomes important to kind of attach, names to these structures. And so these ones that have the internal flagella, right, so they can kind of spin and rotate around. And so again, we don't always have the flagella sticking out on the outside. And so we just kind of want to remember these. So we have the monotricious, one flagellum, polar, they can be on either end of the cell. So it was sticking out, amphitricious, you have one in each cell. Clusters can be on one side of the cell, locotricious types. And then the peritricious, like we saw over on the side over here, can be multiple flagella all over the cell. And so we just kind of want to remember those things. Other kinds of motility we have, again, we're not going to belabor this. We can glide, we can twitch, we can use actin to help us shoot into cells. So the intracellular bacteria, okay, we have invading host cells, right? They're essentially propelled into those host cells. We have adherence molecules, pili, okay, that allow cells to adhere. In the biofilms, that's going to be pretty important to remember. So when we have the cells coming in that initiate the colonies, and we have the cells that are recruited that come in, what is allowing them to, to associate with the matrix and with other cells, okay, within the environment. We have the DNA transfer, the sex pilus, and that's used for conjugation, right? And that's different from, the ones we have all around the cells. And we have these interesting organisms, the Colobacter, Hyphomonas, that have these stalks or hold-fast structures that allow them to stick to substrates. And they can use that also to acquire nutrients. And so in the last couple of things we want to look at today, we have capsules, right? Very important for host immunity, as we saw in the table, and we can stain differentially for them. Okay, so the general bacterial stains, gram stain, acid-fast stain. Okay, so even if you do no other stain, right? Those two you will do. If you want to identify flagella, the capsule, endospores, right? We have special differential stains that will allow us to see that. Okay? So if you wanted, you had a gram-positive cell, you have endospore, you can do the endospore stain, you can see the endospores, right? But the general stains, anybody, anything at all in microbiology lab, gram stain, acid-fast, okay? And then we have these other differential things. One of them is the capsule stain. And so we're looking, at the biofilm, what it looks like, kind of getting us ready for next week. So we have, those capsules, polysaccharides, for the most part, they have sugar components in the polysaccharides. We have, some capsules that are proteinaceous, okay? So the most common capsules, carbohydrate moieties in there. We have some that have protein. But, besides the capsules being important in the host, for avoiding, host immune responses, especially the macrophages and so on. The capsules are also of importance to the cells in biofilms and in the environments where we find them. So they use them, to stick to places, okay? So we have, in the biofilms, we find in the environment, when you see clusters of bacteria on rocks or whatever, you have biofilms over there, okay, on tree barks and whatever. But then, we have the cluster of cells in the mouth, okay, around the teeth, around the gum line, the cheeks, whatever, right? Those are examples of biofilms. And we have the dental plaque, those are biofilms. And as we talked about yesterday, right? So we can have variations in the cells that we find, depending on where a person lives, what they're eating, what's in the environment, and so on. And this is one of the reasons why those who are interested, especially public health, dentistry, research in that area is really interested in finding out if these, these organisms, if they're the same everywhere, or if you have, differences. And if you have differences, what are the differences? Do we have core organisms that we find regardless of where you are, okay? And so what we're seeing over here, if you were to take some material, out of the plaque, from carpet, etc., and if you looked at it, you have, a whole lot of cells in there, okay? In a small amount of material. You can have upwards of, 10 to the 8th, 10 to the 9th cells are present. And so you're looking at the biofilm on the tooth, and then we're looking at the biofilm that we have on a toothbrush bristle. So which means we have a lot of cells we're dealing with. And we have, again, different, we can, gram positive, gram negative, rods, coxine, spiral shaped, everything can be present in that one small amount of material, okay? And so other things that the cells, can use, they can deposit, slime layers, they can have glycocalyx material, structures that allow them to stick. And, and as you know, the plaque can be very tough, okay? If it's, especially if it's been established over time and there hasn't been, good dental care. And so these things can be very hard because we have the production of metabolites, enzymes, acids, and all of these that, are helping to keep things together. And as a result, on the tooth surface, we can begin to destroy that tooth. So again, we can have an appreciation for just how much stuff is present in there. And so we mentioned the S, layers again, very important, structures, and because of how important they are for the cells, right? The individuals who investigate, the uses of nanoparticles are very much interested in this molecule. Okay, this layer, this S layer. And so again, we see that. And so the last, couple of things are the endospores. And so again, we just go right to the mechanism. So again, we have different kinds of spores, okay, depending on the organism. So we can have central, terminal, etc. Depending on the kind of organism you're looking at. So Clostridium will have, different types of spores to Bacillus, okay, and so on. We can see, these structures are important. We talk about sterilization, next couple of weeks. We want to emphasize the reason why autoclaving has to be done properly, okay, with instruments and things like this, because we want to make sure that any spore-forming organism is killed. If autoclave fails, these will vegetate and we'll have bacteria all over again. And so important is this that, people have to keep logs when you have autoclaves. You have to make sure that, they're meeting, the sterilization properties. Okay, so spores cells need to resist environmental insults, right? And then once the conditions get better again, they will vegetate and then you have the cells produced. And so heat, radiation, chemicals, desiccation, all of those things, are protected, the cells are protected. We want to know what's covering the cells inside as we have the spore develop. So that diagram that you have on the top there, please look at the parts of it. So you have the development of the spore layers. So we have the exosporium on the outside, we have the coat on the inside, we have the core of the spore, and then we have, the membranes, inner and outer, right? And then, the germ cell wall. So inside the core, the vegetative aspect of the cell is protected. So when the environment gets conducive, right, we can germinate, and then we have new cells, okay? So, what you see over here, right, again, that peptidoglycan layer, right, is still going to be there within the core, the nucleoid, the ribosomes, all of those things are there. So that when we germinate, basically we have, the cells, dividing, and we have, new cells. So again, those layers are represented over here in the bacterium. And so again, the resistance, we want to know what's in there, the nucleic acid with the calcium, right, the small soluble DNA binding proteins, right, all of these things help to keep the spore pretty resistant. And so to make sure that we can get new vegetative cells developing. And so this thing over here, again, just, we'll just go straight to it. We want to see how the whole process occurs, right? So we want to see, how we form the spore, okay, what happens as we begin to form the core of the spore, right? All the way to when we have the coat synthesized, right? The full coat, and then the spore, right? And so now that in the environment or in the body, when conditions are conducive, we're going to have vegetative cells, okay? So when we autoclave, when we sterilize, we want to kill spores. Because if we don't, we're going to have bacteria all over again, okay? And so, we'll say a little bit more about that, next week as we move on to the oral cavity. But again, this just shows you that, you can activate the cell, you can germinate, and then, the spore foot breaks, and then we have the new vegetative cell that shows up. Okay, and so the cell, again, just regular bacterial cells growing. So those things that we highlighted, we want to make sure that we remember them. For the peptidoglycan, remember, what the core structure is, and why it is, vulnerable to the enzymes that can, destroy it, as well as the drugs that can also destroy it. Okay, and so on Tuesday we're going to begin to look, inside the mouth, look at oral ecology, and begin to kind of lay down, okay, how these biofilms form and what happens inside those biofilms. Okay. Any questions? Okay. So would it be easier if I just sent the notes to you each time instead of sending them to, just in case somebody's away, or tied up with something, you can make them available. Okay.
Yeah.
Okay. All right. Any questions? Okay. All right.